PacBio smart sequencing works by utilizing the natural activity of DNA polymerase to read a template sequence. Smart sequencing has applications in health, agriculture, and biological research, and is one of the most common third-gen sequencing technologies. It works by isolating DNA or RNA. This will create smart bell libraries of topologically circular pieces of DNA.
From here, you will add primer and polymerase in preparation of the sequencing reaction. The reaction will take place in what is referred to as a smart cell. One smart cell can contain thousands of zero-mode waveguides, or ZMWs. A ZMW is a narrow chamber where polymerase will be anchored. A ZMW well is narrower than the wavelength of light and allows light to penetrate the bottom of 20 to 30 nanometers. Inside the chamber, DNA polymerase is at work replicating the template DNA strand. Phospholinked nucleotides with fluorescent labels are added to the chamber. The polymerase incorporates the base into the DNA template, and the phosphate chain is cleaved off, releasing the fluorophore and emitting colored light specific to the incorporated nucleotide. Simultaneously, a pulse of light is created through the bottom of the ZMW chamber, allowing for observation of the fluorescent nucleotide while maintaining high signal to noise. Each of these additions is identified by a sensitive detector in the ZMW chamber, allowing the nucleotide kinetics to be measured in real time.
This process is occurring in parallel within the thousands of other ZMWs located on the smart cell.
PacBio smart sequencing offers no lag time between each nucleotide addition, the ability to handle thousands of reactions, and no amplification step, allowing for lower bias and uniform coverage. One drawback is the PacBio has a lower overall accuracy unless using the hi-fi model. Smart sequencing provides exceptional read lengths without compromising throughput or accuracy. With two sequencing modes, you can optimize your results based on your reads. PacBio is the only sequencing technology to offer highly accurate and long reads, the hi-fi reads, that provide Sanger quality accuracy with the read lengths needed for assembly of the complex genomes. PacBio also allows repetitive sequences to be accurately mapped, simultaneous epigenetic characterization, and the sequencing is done quickly at a somewhat affordable price. PacBio Smart is capable of accurately and rapidly completing targeted sequencing, whole genome sequencing, de novo sequencing, and RNA sequencing.
In comparison to PacBio, Illumina provides shorter reads and higher overall accuracy. It is also restricted to a lower capacity than PacBio. Illumina utilizes linear DNA, whereas PacBio uses circular DNA. PacBio is significantly more expensive than Illumina. There are current limitations when it comes to reconstruction of metagenomic genomes. This paper utilized SMRT technology to achieve a complete genome sequence reconstruction of Corynebacterium simulans, a previously uncharacterized species due to limitations of short-read sequencing technologies. This paper explores the challenges involved with trying to treat HCV due to an emergence of drug-resistant substitutions in the virus. PacBio SMRT is utilized as a way to provide a more detailed understanding of the hepatitis genome. The long contiguous sequence reads that PacBio provides allows us to look at the haplotypes of these infected viral clones. Prior sequencing may have been able to detect these haplotypes, however, is unable to detect low abundance haplotypes, which may have acquired this drug resistance capability.
This paper discusses how smart sequencing can be used to identify many different epigenetic modifications, such as the N6-methyladenosine, 5-methylcytosine, and 5-hydroxymethylcytosine, which cannot be distinguished using other techniques such as bisulfite conversion. They discuss how the properties of each modification affect the interpulse duration, or IPD, detected by the sequencing analyzer. Modifications affect how the polymerase approaches the DNA, as the modifications are typically large, and affect many of the surrounding bases. Thanks to the ZMW, any differences between how a nucleotide is incorporated into the sequence can be detected, quantified, and analyzed.